Greetings, respected judges. I am very honored to present my finding here today. Among the three layers of the skin, keratinocyte lies on the uppermost sheet, making it vulnerable to UV-induced damage, leading to disrupted epidermis and, in the long term, skin melanoma. Now, cannabinoid-sensing receptor has been shown to have a protective effect against this UV damage through its antioxidation and anti-aging properties. However, its mechanism remains unknown. As of recently, GPR55 receptor, a novel class of cannabinoid-sensing receptor, has been deorphanized and to be expressed in keratinocytes. And 01602, a synthetic specific agonist of GPR55, has been shown to have its activation-like phenotypes. This takes us to the objective of the study, to investigate the impact of activation of this receptor on UV-induced keratinocyte damage and its underlying mechanism. Let's begin with the conceptual framework. We used cat cells as a cell-based model, induced UV, 01602 used to activate GPR55 receptors. We look at its effect on cell viability, death, proliferation, and the underlying mechanism. Initially, MTT assay was performed by UV induction, cell treatment, and MTT treatment. In this assay, MTT gets converted by the mitochondrial dehydrogenase into purple formazan crystal. Increase in formazan intensity indicates high mitochondrial activity and cell viability. Now, we found that inducing UV at 25 millijoules alone decreases percent cell viability. However, treatment with 01602 at both 24 and 48 hours of incubation dose-dependently increases that percent viability. However, an increase in viability can be due to reduced death or increase in proliferation. To investigate cell death, we performed the Hertz ethidium homodimer co-staining assay by UV induction cell treatment applying ethidium homodimer dye. The red ethidium homodimer is non-permeable to cells with an intact membrane. Meanwhile, the blue Hertz dye is, so it dyes viable cell blue. On the other hand, in apoptotic or necrotic cells with disrupted membrane, both dyes can enter, and the ethidium dye outcompetes the Hertz, dyeing it red. To recap, blue fluorescence mean living cells, and red fluorescence mean dead cells. In our control group, there are only viable cells, so it stains blue. Now, UV induction alone increases cell death. Surprisingly, though, at both 24 and 48 hours of incubation, our treatment reduces the number of cell deaths. We investigate cell proliferation further as well using the BRDU assay. The standard protocol of BRDU was carried out, and during the S phase of the cell cycle, BRDU, or a higher affinity thymine analog, binds to the newly synthesized adenine bases. We quantify them using an antibody probe with fluorescence. High fluorescence intensity reflects high cell proliferation. As we have hypothesized, our treatment increases percent cell proliferation in a dose-dependent manner at 48 hours of incubation. Once the property of 01602 has been established, it is time to investigate its mechanism. As elucidated by previous assay, 01602 exhibits a positive effect. But this effect was abolished when we added the ERK, SIRT1, PKA, and AMPK protein inhibitors at both 24 and 48 hours of incubation in a similar mechanistic pattern. In the perspective of cell proliferation, similar positive effects was also abolished using the same 4 proteins inhibitor. This helps us conclude that 01602 induces recovery through the PKA, AMPK, SIRT1, and ERK-dependent mechanism. But how do we determine the sequence? Well, based on previous studies in melanocytes, skin cells, and intestinal cells, the sequence is PKA, AMPK, SIRT1, and ERK activation, respectively. So we infer that this is probably the sequential enzymatic pathway. To investigate further, we use standard Western blot protocol to see protein expression. We found that our treatment induces AMPK and ERK phosphorylation. However, that protein expression was abolished when we added the PKA and SIRT1 inhibitors, helping us conclude that 01602 promotes AMPK and ERK phosphorylation by stimulating this pathway. Additionally, we investigate the SIRT1 activity using SIRT1 assay. We measure the intensity at every 2-minute interval, and in this assay, the quencher and fluorophore get deacetylated by the SIRT1 enzyme. The detached fluorophore can be detected using a calorimeter. High fluorescence intensity indicates high SIRT1 enzymatic activity. Just as we have hypothesized, our treatment increases SIRT1 activity, but this effect was abolished when we added AMPK inhibitors. Now, once the mechanism has been established, I must conclude my finding that GPR55 activation induces keratinocyte recovery by reducing cell death, increasing proliferation and viability through the protein pathway of pKa, AMPK, SIRT1, and ERK phosphorylation. Once the in vitro model has been completed, the next step is to study UV-induced melanoma and UV-induced skin aging within a mouse model. Ultimately, though, we want to conduct a population study to see the efficiency and potency, see possible adverse reactions, and conduct drug profiling in a population. Optimistically, I want to develop the product that is able to either treat or protect against skin disorders caused by UV. I would like to acknowledge Chulabhorn Royal Academy, the fundamental funds, and my advisor, Assistant Professor Pravin Pongkorpsagun, for this project. Thank you.
Your insightful presentation was so impressed. Are there anything you would like to inquire about?
What kind of UV that you use for induce?
The UV that we use in this experiment is UVA, which has a wavelength of 320 to 400 nanometers.
So about the inhibitor that we are using for the mechanistic evaluation, how can you know that the drug of target or your substance is not directly interact with the inhibitor that you are using? So in which sequence that you incubate the cell with, and is the result of the inhibitor of them independently, or the interaction of them between your substance and the inhibitors that you are using?
Okay, thank you for the question. So this assay, each inhibitor was induced in our treatment independently. So we investigate the role of each inhibitor independent of each other on the percent cell viability and proliferation. And then we investigate the possible mechanism, which is a sequential pathway, by combining by running Western blot to see the direct expression of proteins from one inhibitor to the other. So this helps ensure that in the first step of the experiment, each inhibitor is independent of one another, and then in the next step where we investigate the mechanism, one inhibitor leads to the reduction of protein expression of another.
So from your first slide that you are just presenting now, I mean in which sequence between the substance of the target or the inhibitors that you are incubating the cell with, or you co-incubating it together?
Okay, thank you for the clarification. We co-treated the substance, so both 01602 and the inhibitors were incubated together with the cell after UV induction.
So to be more clear, do you have a single arm therapy of each inhibitors? If you show only the combinations, do you have
Yes,
single
we
arm
have
of each one,
yes,
like that?
which is demonstrated in this result table. This is a single arm experiment of each inhibitor.
I think we mean that you have the inhibitor only without the 01602.
Yeah.
Oh, we didn't. No, we did not conduct the inhibitor alone assay, just co-treated with the 01602 treatment.
Any more questions for the judges? Thank you very much.